It is my pleasure to introduce Dr. Michelle Dennis. She's a board-certified veterinary anatomic pathologist and associate professor in the Department of Biomedical and Diagnostic Sciences at the University of Tennessee College of Veterinary Medicine. She graduated from Purdue University School of Vet Med in 2002 and completed a combined pathology residency and PhD program at Colorado State University in 2007. Her research has focused on comparative pathology and investigation of natural disease with a special interest in aquatic animals. Present or current research addresses disease in wildlife species of ecological and economic importance, including corals, spiny lobster, sea urchins, freshwater mussels, perinatal hawksbill and leatherback sea turtles, and Antillean manatees. So she's got some really exciting stuff that she works on. And today she's going to tell us about one that is near and dear to our hearts because it's something that we are another biodiversity hotspot, global hotspot for. And I turn it over to you, Dr. Dennis.
Okay, let me get this going. I gotta swap that. Okay, so yeah, pathologist, and much of my focus in my career thus far has been on aquatic animal disease, but I'm actually a newcomer to this mussel scene, taking them up in this last year when I moved to Knoxville and started at the University of Tennessee. So I'm learning so much about freshwater mussels, and what I'm finding is that a lot of the challenges they present when it comes to studying their diseases are not specific to them. I've encountered a lot of these challenges with marine invertebrates. So some of the themes are the same, but then also some of their challenges are unique, and they're all interesting to talk about and how to address them. And so I'm going to focus this talk on the challenges, but also put it in the context of what we're doing to address them through the study that the One Health Initiative seed grant funded. And so in doing that, first up, I just wanted to mention that while I'm the one talking, I'm just a small part of a much bigger team. And so if you look at the appointments of those on this team that I'm working with to study mussel decline in our region, you'll see that, let me just get my pointer going. So we got a couple pathologists, we have a malacologist, fish ecologist, mathematical disease modeler, microbiologists and wildlife health specialists, river restoration ecologists. You'll see that these disciplines, some of us, we don't often come together. We really had to take a One Health transdisciplinary approach to tackle the challenges that I'm going to talk about. And last but not least, I also want to acknowledge Geronimo Silva up front, very talented and bright PhD student sent from heaven that we're really lucky to have kind of doing a lot of the work on this project. Okay, so can you see in this photo, the One Health poster child of Tennessee? Well, I could go bigger and even say One Health poster child of the US Southeast. Did you spot it? How about now? So what I'm showing you are some mussels in the Clinch River. So mussels are a special part of our local biodiversity and natural history. They're a part of our culture, part of the culture of our region, part of what makes our region special. So they're spending most of their lives, which is about anywhere from 10 to 100, even 100 years, with their heads buried in the bottom of the river. They don't look like they're doing much here, but they have a super important role, which makes them this One Health poster child. And I'll tell you about that role in a minute. But first, I wanted to show you this. So this is a female, I'm told this is a female oyster mussel. And so what she's doing is displaying here on the left. So this female is gravid. She has these pouches within her gills, which are filled with her offspring, her babies. So her larvae are called glochidia, and they're in these pouches. And she's displaying a lure here at one end. And that lure is made from her mantle, the soft tissue that's lining the inner part of her shell. And it is mimicking an insect larva. that the intermediate host, which is going to be host to her Glochidia, prefers as prey. So what's going to happen is that intermediate fish host is going to come up to this larva, and unexpectedly, she's going to surprise this fish host and deposit her babies on its gill for them to develop completely. Isn't that neat? And here's something else that's really neat that they do as part of reproduction. So some mussel species, instead of a lure, will produce a conglutinate. So this looks like a little worm, doesn't it? But this is actually a packet of a female oyster's larvae, which again is mimicking the intermediate host, what the intermediate host prefers to prey upon. So something like an insect larva. And once that fish ingests this conglutinate, then it's going to break up and all the Glochidia will be released and they'll parasitize the gill. So it's got these freshwater mussels have a fascinating life history. So this is just kind of summarizing what I explained to you, where the female oyster is making a display that's going to lure in the intermediate host fish species, and then those Glochidia are going to get deposited on the gill, and then they're going to develop into juveniles and drop off and burrow into the river bottom and then develop into mature mussels. They're so cool. But wait, there's more. So freshwater mussels, we consider them as keystone species for river ecosystem health. And for many reasons. So first, as they're buried in the bottom, sometimes in great numbers, they're stabilizing the riverbed. They're also important food sources for many species of wildlife: otters, raccoons, muskrats, herons, egrets, some fish. But the most important thing is their role as filter feeders. So what they're doing is constantly siphoning organic matter from the water column. So that's the filter feeding. That in this water, they're going to take up algae, bacteria, silts, other particles, and they'll be purifying the water as they do this. But then they process that and they're going to excrete nutrient that gets used by plants or gets deposited as organic matter in the sediment for invertebrates and fish to feed off of. So they're at the very bottom of the food chain. Everything in this ecosystem above them is depending on this nutrient cycling. So we could consider these guys our Brita filters and our river feeders. So here they are again, our ecosystem engineers. They are the trophic level that matters the most for a river basin ecosystem health. When they are lost, it's going to have indirect impacts for all end users of that ecosystem. So this is why they are such a great One Health centerpiece, great One Health story. And unfortunately, it comes with their unique life history that they are considered to be highly sensitive to environmental stress. So many of us living here in Tennessee already know that we're in a very special area of the country, some could argue even world, and that we are in a biodiversity hotspot. But sadly, we are also in an imperiled species hotspot. So this is a map that's showing imperiled species locations, that is species that are on the Endangered Species Act or otherwise considered in danger of extinction. And so here you can see Eastern Tennessee lighting up like a Christmas tree for richness in imperiled species. And this website was really neat. It allows you to apply a filter to say, okay, well, how much of this imperiled species is in aquatic invertebrates? And so here we can see aquatic invertebrates topping the charts. So these species are a big part of what the story of imperiled species in our region, and a big part of this is freshwater mussels. So this graph is showing you, well, a few things. First, start here. This is showing us in North America how many freshwater mussel species we have compared to other parts of the world. So this is showing us that we kind of have a special gig going. We have the most freshwater mussel species. But then if you look at these bars, this is showing you different threat levels. And you can see that while we have a lot of mussels, we're not looking after them very well because we have, according to, this is from Nature Conservancy numbers, 70% of our mussels are extinct or imperiled. So that's higher than other countries, and that's much higher than what's happening with mammals and birds. So freshwater mussels are really under pressure. So what's happened? They've dramatically declined throughout North America over the last 1.5 centuries. For some reason, mostly not understood, although we know that the way we have used our use the habitat, so the extent to which the habitat has been modified, especially in terms of placing dams and impoundments, that's had a huge impact on mussels. We know that much. We know there's been historical pollution, and invasive species are an issue in some areas. So kind of the bigger black box comes in more recent decades, where we've had epidemics of mass mortalities throughout the country, well, throughout the world really, which are increasingly frequent, and they kind of have the same theme in that they've been poorly investigated and poorly understood. So this just coming out in the news a couple days ago, U.S. Fish and Wildlife Service have delisted 23 species from the Endangered Species Act. We've given up on them, and we're just going to go ahead and declare that they're extinct. And so I was looking through this list, and here we have 6 species of mussels, and 5 of those are in Tennessee. So here it is happening, right? Extinctions happening in our lifetime right in front of our eyes. These species never to be seen again, lost from Earth forever, for reasons not entirely understood, but on our watch. So we are in dire times. We have hatcheries in Tennessee, Alabama, Virginia, Kentucky that are working to restore these critically imperiled Tennessee fauna, freshwater mussels. All right, so this is showing you kind of a beautiful area of Tennessee. I'm gonna focus specifically going from big picture mussel declines to what's happening in Tennessee now. So this is showing you a view of the Clinch River. So it's located in northeast Tennessee. It's flowing down from southwest Virginia. And this is one of the, well, it is the most biodiverse river system in North America. And mussels are a big part of that story. So it harbors 46 species of mussels, nearly half of which are endangered. It's considered one of the few remaining havens for North American freshwater mussels. So a very special place. So sadly, starting in 2016, biologists started to observe mass mortality in this Clinch River. And so this slide is showing you an early news story about initial observations where there's a biologist and describing finding over 1,000 dead mussels on the site. And unfortunately, it was not a one-off event. It has been observed for several years now. It tends to have a kind of seasonal occurrence. And there's still a lot that we do not know about why these mussels are dying here, and even what species are involved, what ages or size classes of mussels are most susceptible. So one of the co-investigators I'm working with, Jerry Dinkins, helped with this slide. He labeled all the different species he could identify, and it's really kind of, I don't know, I just feel like it sends a strong message when you look and you see so many of these species here in this one photo are federally endangered species. So it's thought that some of the species in the Clinch River have declined from 40 to 90% since these mortality epidemics were first observed. So this is just a map of the area where these mortality epidemics have been seen. And so Kyle's Ford, that was, I guess you could call it ground zero. This is where we go back and look at these news stories. This was Kyle's Ford, where this mortality was first observed. And then soon afterwards started to see mortality at downstream sites. And so that led to the belief that there was likely a contaminant or an environmental factor that was the basis for the mortality. And I'll tell you on the subsequent slide a little bit about the checkered past of this region upstream in the Clinch River. So most initial investigations were concentrating on contaminants, but then subsequently mortality epidemics were observed in the following years upstream. And then so that led towards a shift in attention towards infectious factors. So other things that we know about what's happening in the Clinch River is that it seems to be most prominent during the stressful time of seasonal turnover period. So there's a lot of temperature change. It's stressful. And there's also impression that it's not as bad in years that have had like a lot of rainfall. So wet years, the mortality does not seem to be as bad. But these are just kind of the impressions. So this is the kind of more background to the story, looking at what's happening in the upstream Virginia side. So this red area called the zone of decline, this is where mussels have decreased around 70% in density from the late 70s to the early 2000s. They decreased 70%. And so this area, the historical declines are associated with agriculture, mining, urban development in the surrounds. And there's been incidences where wastewater effluent has been documented or implicated, chemical spills, agriculture runoff, deforestation, all these sorts of things, which could have bad effects for the mussels in that area. And this study has studies have shown that mussels that are grown in this area do not survive as well. They do not grow as well. And this study has shown that mussels grown in this area, the zone of decline, they have higher levels of polycyclic aromatic hydrocarbons than mussels not grown in that area. And also that the levels that they're detecting in the tissues of the mussels are negatively correlated to mussel density. So it seems like these PAHs that are related to, or at least thought to be derived from historical coal mining, are at least playing part of the role of the declines in this area. And definitely an important environmental factor to consider for stressing the host in the region more broadly. Okay, so stepping back away from the Clinch. The Clinch is a very depressing story, especially because it's such a treasured biodiversity hotspot, but it's not a unique story by any means. So it's very similar. The story that I just told you is very similar to what's happening kind of throughout the United States or wherever mussels are globally. So we've been seeing these episodes of mortality that are associated with declines, and they're occurring in locations that do not have an obvious explanation for mussel decline. So there's no obvious environmental impact that the decline could be attributed to. And other aquatic species in those habitats are not affected. It just seems to be mussels. And so authors have been using this term enigmatic to explain, to describe these mussel declines, because they're there for no evident reason. And they're happening after we've had these major laws that have been passed to protect the environment. So, in the past, we have had these obvious episodes of pollution, but we don't have that anymore. And now we're seeing these episodes of mortality, and the declines are continuing and we can't explain it. So they're enigmatic. So when talking about wildlife population declines, we could think of it from just a very simple kind of population growth model, that there's going to be one of two things happening. Either the birth or recruitment rate is decreasing. And when we think about that, we could think about habitat loss. There's just not space to sustain animals coming into the population. There could be, with mussels specifically, that they don't have the right fish hosts to complete their life cycle. or there could be a reproductive disease affecting the mussels so that they're not reproducing in the first place. So there's either there's a decreased birth and recruitment rate, or that there's an increased death rate. And top of the charts for increased death rate, especially if you're observing increased mortality, would be disease. Very rarely do we have environmental conditions that are just not compatible with life whatsoever, resulting in acute death of animals, but it does happen. So like if there's complete loss of dissolved oxygen, pH is not right in the water, extremely high levels of contaminants or something along those lines, then we could have episodes mortality. And then, you know, trauma or predation, that could result in an early death of an animal without disease as well. But it's hard to think of examples of that contributing to declines in the wild, maybe like with lionfish in the Caribbean being voracious predators, introduced species that have a voracious appetite. They could probably contribute to declines. So when we think about disease becoming increasingly an issue to the point that it's causing a decline in a population, it's really helpful to consider it from the point of the view of what we call the disease triangle. So that's this figure here. And this is a concept borrowed from plant pathology, but is really applicable in animals as well. And I think we've even seen it in other One Health seminar series talks. So here it is again, and this concept, basically the story here is that if we're going to see disease, we have to have a virulent agent, a susceptible host, and a favorable environment. So when all of these things converge, we can see an increased occurrence of disease. Or conversely, if we have a weaker agent, a stronger host, and a non-conducive environment, then we'll see less disease. So it's helpful to think even with mussel declines from this perspective. But again, disease is a big part. That's something that is deserving focus when you're dealing with a decline issue. And I don't think I'm just being a biased pathologist by saying that. So in this paper that was reviewing the enigmatic mussel declines in the US, there was this great table that summarizes the conventional wisdom. So the author is saying, you know, these are factors that people often implicate, but he recognized that many of these are very circumstantial, and there's often little, if anything, ever done to substantiate any of these claims. And here we have disease at the bottom, needing investigation. So when I look at this list on this table, I see a lot of things that would contribute to a lack of recruitment or habitat loss. And I see many things that are environmental issues, not environmental conditions that would be like extremely incompatible with life suddenly, but many environmental issues that would come to play in this disease triangle, things that would make a host more likely to develop disease from one mechanism or another. In any case, disease is rarely investigated. We're starting to do it, but we find that there are many challenges to get the same kind of answers that we're used to getting when investigating disease in terrestrial animals. And so I want to go through those challenges. And before I do that, I need to get on the same page with you about what's involved in a traditional biomedical approach to investigating disease in a context of a wild animal decline. And so what I'm going to do is use the bat as an example in the One Health spirit. And I'm doing white nose syndrome caused by Pseudogymnoascus destructans, a fungus. And so this is a disease that emerged in 2006 in upstate New York. So not that long ago, but within a decade of its discovery, it killed 6 million bats, and it has caused significant declines in multiple species. So this is a great example of a single disease causing declines, and why it's worth looking for disease in the context of declines. Okay, so what do we do when Well, first of all, we need to know there's a decline, and that can be hard sometimes depending on the wildlife species. We might need surveys or active surveillance to detect it early. It may get to the point where the public is observing sick or dying animals before you know that it's happening. Then that'll instigate a field investigation. And what this involves usually is describing the extent of mortality, how fast, if a disease is causing clinical signs, how fast it progresses from illness to mortality, the percent of cases that die, who the susceptible hosts are, if they have specific factors associated with susceptibility. Age would be a common one. Gathering any other pertinent historical information that might give us some clues about what's happening. Next, for often coinciding with this, we do physical examination of a sick animal. So here we have a close-up of a bat with the white nose syndrome. It's got these white plaques around its nose. But in addition, we might do some blood work, diagnostic imaging, other tests, trying to hone in on what is going wrong in the animal, what body system, what tissues are not functioning properly, what's going wrong that's leading to death. Whenever there's mortalities, there's definitely going to be postmortem examinations. And so this really is an obvious advantage in terms of comparison to doing tests on live animals where we're trying to get at what's happening in the body. Well, here we can actually just open up the animal, look at the tissues, and see for ourselves which ones aren't functioning, see the abnormalities either with our naked eyes or with the help of a microscope. What is the abnormal process that's happening, and what are the most likely causes given that process? So here we have a couple of bats. This one's opened up and it has a hemorrhagic enteritis. This one that has some plaques on its wings. We're carefully examining the tissues for any kind of abnormality. Now, when we select animals to do postmortem exams, and usually depending on the scope of the mortality investigation, there'll be quite a lot of this because the amount of information that is produced is very valuable. But when we're selecting animals, it's really key in terms of picking the animals, that we want the ones that present the most clear and uncomplicated view with their pathology, the abnormalities that we identify. We want an uncomplicated view. Now, the problem is in populations of animals, they're typically not all animals are healthy at any one given time. There's often things in the background. So background diseases, background infections, which might have a low level of mortality or no mortality at all, just there. But that can complicate the issue. So we generally don't focus on pre-symptomatic animals. We want animals that have become acutely affected. They just started showing clinical signs. If we wait too long, then we might start dealing with secondary issues, or we might see changes in their tissues predominate that just reflect that they're sick and trying to heal. And those changes obscure what initiated the injury in the first place. So have you ever gone to the doctor starting with a cold and then you didn't get better and they end up putting you on antibiotics, classic primary viral disease that ended out with secondary bacterial infection? We don't want to look at the animals and see the bacterial infection. We want to see the thing that started the injury. We want to diagnose the viral infection that's the main cause of the problems. So that is why we want acutely affected animals, and why selecting the animals to do this investigation is so key. And I'm going to come back to that in this talk. The next step is we're going to do a microscopic examination of tissues. So here we're looking at the bat skin, and we can see that there's a dermatitis here, and associated with that is this fungal overgrowth. We have these hyphae and arthroconidia at the surface. So right away, we've ruled out a lot of things doing this step. We can tell you it's not toxic, not bacterial, not parasitic. It's not an age-related thing. It's not a nutritional related thing. It's fungal. So we've narrowed down the list of possibilities, but it could be a lot of different types of fungi. And so the diagnostic tests will be aimed at that most likely cause and figuring out exactly which agent it is. So with white nose syndrome, we actually started out with a metagenomic approach where they identified all these different fungi. They also cultured a lot of bacteria in the beginning, and it wasn't without histology that we could say, these bacteria are just secondary. With the fungi, it took careful examination of the tissues in comparison to isolates and culture to narrow down which fungi were actually the most likely cause. And it turned out that the P. destructans wasn't even in the list of likely candidates that was metagenomically identified from the start. So the ultimate step is an experimental infection model or exposure model where we prove we think we found the thing that's causing it, and now we expose naive animals, and that incites the same disease with the same morphology of abnormalities under the microscope, and we re-isolate that agent. That's what we like to do. Sometimes we do population studies to make kind of like build our epidemiological evidence, short of doing an experimental model. So to do these steps well, it really requires an understanding of how a host manifests different types of injury. And it really helps if we have an understanding on what these background issues might normally be in a population. Things that are going to be there, kind of like red herrings that might get our attention but aren't really causing the problem. It also really helps us if we know what their microbial flora are, or even pathogens that are normally present without times of declines or mortality epidemics. And often in wild animal populations, we're not doing that type of surveillance, and we don't know until there's a problem. And then we got to kind of backpedal and try to figure this stuff out. Okay, so now, how do we go about investigating diseases and these little guys who are living their secret lives in the bottom of our precious rivers, doing a really important job, and we don't want them to die? 7 major challenges. I'm going to go through 7 major challenges. The first problem with aquatic animals is that it's all happening under the surface and not in clear view. So the first major challenge is observing their illness and mortality before it's at disaster levels. So whenever we see any aquatic animal dying in mass numbers, we are really seeing the tip of an iceberg. And it's more so when we're dealing with a cryptic species, or one that's not in clear, that's less likely to be in clear view, such as a mussel that's burying itself. We really want early detection because, especially with endangered species, we know that if we're going to figure out the cause or the basis for mortality, it's going to involve sacrificing animals. And you can actually get to this point where you do not want to sacrifice the critically endangered species to figure out what's killing them. It's hard to convince everybody to take these biomedical steps that we do for diagnosing disease, and we get in this position where we just slowly watch them, the last numbers fade away and die. So definitely want early detection, but active surveillance is expensive and not practical. So here I have an example. If you really want to do like a full survey for mussels, we got this quadrat in the water. And so Geronimo, our PhD student, was explaining to me, what's all involved, that you're going to be digging down 6 inches deep, trying to keep all of the layers on the river bottom intact. So you're not trying to scramble everything up, but you're digging down, moving every mussel if you want to get actual counts. Short of that, you're going to be doing a lot of swimming, looking for any dying mussels. And then if you're doing the swimming, you really have to nail it on the timing. You got to catch it when the mortality epidemic is underway, or we can't do the field investigation. So in other aquatic animals, you're just kind of out of luck, but luckily with mussels, there's a bit of a silver lining, and that they leave their shells behind after they die as evidence. And so here's Jerry Dinkins at the McClung Museum of Natural History and Culture, and he has compiled a collection of several thousand specimens from these mussel mortality events in the Clinch River. And they represent 33 species, 15 of which are endangered. And so the hope is here, well, he's got them all sorted by species, location, and by date. And what we want to do is to take a look at these carefully and hopefully identify size classes or age classes or species that might be relatively more susceptible to whatever's going on in the Clinch. So if we look at their sizes over time by year, if the average size is getting larger, for example, then we know that the smaller or younger mussels are being harder hit. So even basic information about which species are the hardest hit can tell us a lot of clues. So like infectious agents would tend to be host specific, or depending on the natural history of the certain species, they might be more prone to be exposed to certain causes versus a contaminant or something environmental would kind of hit all species equally. But also it's important to know which host species to focus on. If we're going to be studying the mortality, we want to be studying the right species, one that's highly susceptible. And this part of the study funded by the One Health Initiative will also help us understand the population impact that these mortality events are having. Okay, so next on the list is we need to accurately be able to categorize health status, but that is really hard in mussels. So normally we would do a physical exam to find the sick ones, the ones that had just started to show clinical signs. Okay, so you try to do a physical exam on a mussel and they're just closed in their shell and you can't get to them. So when mussels get sick, they basically do the following. They stop moving around as much. They may be found unburied on the bottom of the riverbed. They might not correct themselves when you put them back into the riverbed and appropriately oriented. They might be gaping. So that's next level. And then if you touch them, they could close up, or if they're really sick and you touch them, their foot's just going to be flaccid. They don't move, they don't close up. Then they're really sick. That's when we would consider them potentially moribund, and then they die. So not a lot to work with here. We really can't, it's not practical in the field to observe mussels for, half an hour, see if they're moving enough. Sometimes it's even hard to tell if they're moribund versus freshly dead, and that could really confound results if you're trying to isolate bacteria, for example. You don't want to grow a bunch of bacteria that are just overgrowing the carcass post-mortem. So that could really confound results. We've already talked about why being able to classify kind of sickness versus non-sickness is important for targeting the right animals to do your mortality investigation on. But then think about it from kind of a population level. If you're trying, for example, to find host factors associated with illness, or isolate microbial organisms that are associated with illness. If you just wanted to make a simple odds ratio, the odds of having this mussel disease is higher if they were exposed to this virus versus not exposed, well, then you need to be able to accurately classify if the mussel is diseased versus not diseased. So this one, gaping, I guess we could put it in the disease category, although this can also be a stress response that they recover from completely. And like say, for example, it was just a really hot day. Then what about these ones that are just closed up? Well, your guess is as good as mine. They could be healthy, but they could also be very sick and just not gaping yet. So pretty much any kind of statistics we're doing that's classifying cases and controls based off of health status, just looking at them is not going to be accurate. We have a great need to for more tools to help assess their health. And we can find studies out there where people do neat things in an experimental setting, for example, watching the rate of cilia movement or particle uptake, or using infrared sensors to detect a heartbeat, but there's nothing at this time that's really practical in a field setting. But someone could really dedicate their career to this and move the field ahead milestones. So there's a need for health assessment and diagnostic tools. So right now we're left with really non-sensitive things to classify health, like watching their growth over time for, a long time. If they grow more slowly, well, they're less healthy. Or if they don't survive, well, obviously. Even looking at their condition is really hard to do. So in other aquatic animals, we might take the length and the weight and do a ratio, but we can't do that with mussels even. You can weigh them, but it's really inaccurate because they take a lot of water into their mantle cavity. And so that water is not in a consistent volume. It can vary. And so it's really going to confound any kind of weight results you get with each measurement. So even that is a tough one. Hemolymph is getting a lot of attention because it's a proven technique. Here we have our PhD student Geronimo taking a hemolymph sample from a pheasant shell mussel that was moribund. And this is a great sample because it's sublethal. There are studies that show you can collect so much volume from a mussel and it's going to survive just fine. So the hope is that we can take the hemolymph and we can evaluate it for hemocytes, enzymes, ion levels, and there are studies that show this is possible using our same veterinary techniques that we use in terrestrial animals. We can do this on mussel hemolymph, which is their equivalent of blood. And so through this study that was started with the One Health Foundation seed grant, we were able to also get funding for a pilot study from the Morris Animal Foundation, where we're going to see if any of these analytes that we're looking at with hemolymph assessment associate with disease status defined by the pathology assessment, and then see if any of them are accurate predictors of healthy versus diseased mussels and can be used in the context of a mortality investigation in that way. And now in the omics, the dawn of omics, there's so much that we can do that deserves being looked at: metabolomics, transcriptomics, proteomics. Are there any markers out there that can accurately classify the health of a mussel? And this is going to become increasingly important, not just for mortality investigations, but for monitoring mussel health in the wild, early detection of mortality, predicting mortality, just identifying stressful environmental conditions. And then, from the context of restoration and growing animals in hatchery and then translocating them to a wild, screening their health in a way that's sublethal. Okay, another major challenge. For me as a pathologist, this is the most frustrating one. You finally get some histology samples and looking at the tissues of these sick and dying mussels with the microscope, and I'm finding abnormalities. And if this was a mammal and I see an abnormality in the tissue, I'd be able to give you a short list of known causes that could result in that morphological tissue change. But that is not the case in invertebrates because they've been poorly studied. So it's kind of a hangover of doing so little disease investigation in invertebrates that now we are left it's like we're in the dark ages. It's exactly like we're in the dark ages in people when it was forbidden to do postmortem examinations. And then we just start doing them and we're going to learn a lot and see a lot of things that we don't understand. That's where we're at with mussels. So here is a kidney of a mussel and it's got this conglomerate of hemocytes within the lumen. What does it mean? What's causing it? I have some ideas, but basically everything is an assumption that I'm extrapolating from what I know in vertebrates. Here's a mussel that has a hemocyte infiltrate, same thing. And now my PowerPoint has froze up. Oh, here we go. Okay, so how to get out of the dark ages. So you might think, well, then this isn't going to work. The traditional biomedical approach won't work. Yeah, it's a time-proven approach. The problem is that we're kind of like in a race. The animals are dying, and now there's so much that we need to learn. And it's going to be hard to learn it quickly. But the answer is, instead of not doing it, it's quite the opposite. We need to do it more, a lot more. We need to document the lesions that we observe associated with disease, with contaminants, other environmental stressors. It might be necessary to experimentally induce these things in aquaria to help us interpret what we see in the wild with natural disease. The whole field of omics offers a lot of opportunity. So we're kind of ahead of the dark ages with this because now we can say, aha, this specific change in the tissue is associated with these certain pathological pathways or physiological pathways in the host being activated. That means that this is most likely an inflammatory response, or this is most likely associated with aging, or this is most likely a healing thing. It'll help us interpret it. So we need to do more studies like that. And then, of course, lots of communication and learn from each other as we're going. So along the lines of limited understanding of how a host responds, we've got a lot of limited knowledge on their pathogens. So this is from those first moribund pheasant shells that we took out of the Clinch. And sure enough, they have pathogens in their tissues, but what does it mean? So this is the digestive gland of a mussel. It's equivalent of a liver, and it's got these intracytoplasmic bacteria. So these are rickettsia-like organisms. And here is a ova, and it's got this protist in the cytoplasm. Neither of these, do a search, literature search, have yet to be reported. Is this just part of the background of what's happening? Like, have they been there all along, or is this something new? So the limited knowledge on pathogens, limited knowledge on how the host responds, those are two critical things that we said makes our whole basic biomedical approach go much more smoothly. And here it is, a bit major challenge. So a lot of the techniques that we use for isolating pathogens are extrapolated from mammals. And so there's a great need to optimize culture methods for detecting bacteria and viruses in the tissues of mussels. Not just for a diagnostic, but say, for example, we decide, this virus is the most likely cause. Well, then we're going to need to be able to propagate it in the laboratory so that we can refine diagnostics or study the virus, experimental trials, ... So new advances in sequencing technology and computational biology have allowed us to take it to the next level, where we're not just isolating single pathogens anymore. We're not just limited to that, but we can look at the entire microbial community as a whole. But then it's kind of a double-edged sword, because we have all this information, and what does it mean? Now you have to interpret what that means. And so there's a great need to have more baseline information from healthy populations on what their pathogens include, like what's normally there. But the problem is we're running out of healthy populations. So some great scientists doing wonderful work in this area, addressing mortality in the Clinch, making some really neat findings. And I just wanted to visit some of them in the context of these challenges, not as a criticism, but saying, we are making an advancement here, but this is what we're up against. It's really hard. So lots of respect to all investigators working in this field. So here in this study, they found this bacteria, which occurred in relatively high prevalence. So 40% of the pheasant shell mussels had this bacteria, Yokinella, during the mortality event. But in February, a few months later, it was not isolated. And it was not isolated from a non-impacted site. So that is really provocative, what is going on with this bacteria? So one of the drawbacks in the study was that they were using conventional culture techniques, which we said, we really need ones that can grow all the things from mussels. But they took it to the next level and looked at the metagenomic approach at the microbiome, and there it was again, the Yokinella. So it says it was in higher abundance in the cases than the controls. But there it is, that challenge again. So the case was a moribund mussel lying on the substrate gaping. And the controls is, well, a mussel that didn't look diseased, but we already know they can not look diseased and be pretty sick. So still great information, it's baby steps here. So they showed that they have higher bacterial loads and shifts in microbial communities in moribund mussels. So now we need to run with this lead. Is this just what a sick mussel does, or is this bacteria showing a role in disease? So as a pathologist, I really want to see a linkage with what's happening in the tissues of that organism at a microscopic level. Show me that gram-negative bacteria in the tissues causing an abnormality, and I will be so excited. So another study where they identified, same thing, a metagenomic approach, but not focused on bacteria, but on viruses. And they found this neat densovirus that hadn't been described before. And it was 11 times more likely to be found in the cases than the controls. So what's going on here? Is this an opportunistic pathogen? Well, we have limited knowledge on the pathogens. We don't know how many densoviruses are normally kicking around in mussels without mortality epidemics. Is it the cause? Or is this just, an opportunistic infection associated with a moribund state? What I would like to see is that virus in the lesion causing the problem, show it to me using in situ hybridization, immunohistochemistry. We have these methods to actually show it in the tissue. Or if we can't do that, an experimental infection, but we need the methods to cultivate the organism. So you can see lots of challenges. So what's missing? Solid understanding of what these agents are normally doing in mussels that aren't having mortality epidemics, and linkages, better linkages with pathology. So here's another moribund mussel we got out of the Clinch River, and this is looking at some connective tissue and a hemolymphatic space that is filled with hemocytes that are dying. And they have bacteria in their cytoplasm. I really want to know if that's Yokinella, if it's gram-negative bacteria. To figure out who that is, we have to have other techniques in parallel, either culture it, identify it with microbiome, or use one of these in situ hybridization or immunohistochemistry techniques. This is showing you a similar scenario, but this was a mussel from a different river for a different project in Tennessee. But this is linking histology to microbiome. So we had histology on some mussels showing degeneration of cells in their digestive gland. And if we looked at them closely, you see these tiny bacteria that were gram-positive rods. Well, that's not very common. And those mussels that had that lesion had a relatively high amount of one certain bacterium that fit the profile, a microbacterium, which is something I never encountered in other species. So we need to do more of this kind of linkage of pathology with other tests. And it's hard to do because they're small animals and not a lot of tissue to work with. So to help overcome some of these issues, with the problems of differentiating sick versus healthy and understanding the pathogens, what's specific to mortality versus just a background problem, we set up with the funding from the One Health Initiative Seed Grant, a longitudinal study aimed at linking the pathology and microbial isolates to the observed mortality in the field. So we sourced pheasant shell mussels from a hatchery in Virginia. We had a lot of debates about who to use as our little sentinel species, but in the end, it was deemed this was the best approach because they were considered least likely to be complicated by background disease issues. And other researchers were using this species as well in the area, so it seemed like our results would be the most comparable. So we put them in silos. So silos are a well-established way of following individual mussels over time. Otherwise, if you just put them out there, you'll never find them again. And so this approach has been successfully used by other investigators looking at enigmatic mussel declines, but mainly looking at things like growth and survival, or monitoring them for acute periods of mortality, or exposure and accumulation of contaminants. So the mussels are in here in this little cage in the center of the concrete. Here they are. So we had 100 mussels that we deployed at 2 different sites in the Clinch River. Here's the team. We have Geronimo, Jerry, Gus Ingman, Justin Wolbert going out there doing all the hard work while I'm sitting on the edge eating snacks. And so here they are placing the silos. We did 10 silos at each site, and we check them. Right now we're checking them twice a month, and we're sampling 10 mussels once a month. And I didn't mention that some of our silos at each site have hobo loggers to monitor temperature over time. At our health checks, we're measuring the growth of the mussels and obviously looking for survival or clinical signs. When we sample the mussels, we collect hemolymph to do the clinical pathology assessment, and then also to evaluate its microbiome. And then we'll do a postmortem examination complete with histology and microbiome of digestive gland. And so it's hoped that as we sample before mortality, during and after, we can start to tell a story about what's specific to the time of mortality. So here was what the Clinch looked like a couple months ago, near one of our study sites, looking really green, so nutrient rich. And when there's a lot of nutrient in the water, that can lead to algal overgrowth. And this is just a nice view of, oh, we cannot forget the environment in this context. It's going to be especially important confounder for studying mussel mortality. Probably important driver. So what could go wrong in the environment besides, this sort of scenario? pH, well, when algae boom, they often bust and then they decompose, and it takes up all of the dissolved oxygen in the water, makes it anoxic. So dissolved oxygen levels, pH, suspended solids, contaminants, flow. There could be biotoxins from algae or cyanobacteria, so many things, and that's just the water. Then we have to talk about the sediment as well, which is important to mussels too. So we're planning to look at the temperature and flow and how that's relating to what we're seeing with the growth, mortality, survival, pathology, microbiome. But then, short of, you can't address all of these things at once. So short of that, our team is also going to be taking a mathematical approach. So this will be led by Nina Pfefferman. So we're going to look at population viability and contrast different predicted scenarios. So for example, if it's a contact-based infection, mortality should look like this in terms of how it's spreading. Or if it's a river flow-based contamination, it would look like this. Or you could even, try at, like, if it's a vertically transmitted, how that might look like, for example. So I've been talking all about this category of death because we are dealing with obvious mortality, but declines can certainly have an element of birth or recruitment issues. And to my knowledge, nobody's looking at the pathology of larvae and glochidia and what's happening with them. So that's a whole nother story for another time. We could do probably another seminar on that. So this is a big problem. It's not going to be easy to address, but if we care about our rivers, it's something we very urgently need to address. So every species we lose is an irreversible mistake on our part. And I've just really been inspired at how this group came together and how much help we've had. Really big acknowledgement of One Health Foundation, the One Health Initiative, for making this possible. And also all the co-investigators involved with this project. And Jordan, Richard, Tim Lane were especially instrumental in planning out the projects that we're doing and implementing them. And also Rose, who's been a great help in implementing the project in the Clinch. So I think I have some time left for questions.
That was great. Yeah, does anybody have any questions right now? You can, I think Alyssa will help you unmute. You can type it in the chat or.
Yeah, everyone should be able to unmute themselves now if you have a question, or you can also use the chat.
Perfect. I'll ask a question. So there is a great need, a huge need for just people to start looking at this and acknowledging it and dedicating their careers to this. What do you think is the biggest obstacle? Because we see this in a lot of areas. Corals are another one. And so what's the biggest obstacle, do you think? Why are they not flocking to this? Because I tried to get students to flock to it.
I think I can speak from the pathologist point of view that it's a frontier. I can't reach for a book and just look at when I see a finding that I don't understand in the tissues of a mussel or other invertebrate. I can't reach for a book, look up that tissue and get a list of potential causes. The books have yet to be written. And so some days after looking at a bunch of coral slides or mussels, I just want something really easy, like a hemangiosarcoma. I just want like something straightforward. So I think it's a little bit demoralizing being on a frontier and constantly not having the answers and needing to forge ahead. I think that's kind of one of the bigger areas that gets people not into the field. Also, as a vet, working as a pathologist, I'm working mainly with vets who understand what I do. And I've found that a little bit challenging in the past with unusual species who haven't been working with vets and they don't understand what I do and what is to be gained from pathology. So there's kind of like an upfront marketing that has to happen to explain exactly how this is going to contribute to solving the problem. And it's easy when we're in our own disciplines to forget, it's easy to forget that others don't understand kind of all of your ins and outs of your daily life. It's easy to take that for granted. And so, I guess one answer to your question is it's the One Health paradigm that, is really bringing us together. Without it, we're just staying in our own little disciplines, working on our own little things and not addressing challenges that require people that don't usually work together to come together, take that time to communicate about what it is that they actually have to offer and how it's going to work and what it's going to look like.
I agree 100%. Any other questions? I'm going to ask one more.
Okay.
So when you see, because this is a big question I always have. When you see the bacteria primarily in the bloodstream, but you do kind of see it a little bit in the tissues maybe at death, and you see it in the cells, do you think it's hard to know the significance, as you know, because I often wonder, is that bacteria what ultimately caused the death and how? Or is it something that the animal's dying and it's moving in, but it's still alive enough to start fighting it off a little bit? They kind of die like turtles do, it's like this.
Like a slow drawn out. Yes. Yeah. And so I think that's probably what's happening is that, and I believe that's what in that most recent paper from Jordan Richard and that group that was looking at the microbiome, I think if reading the bottom line, I think that's the hypothesis, is that these are the final blow to the animal. These are secondary opportunistic microbes that are following on this, tails of something else. And it's that something else we really need to get at. So it looks like they're dying from a bacterial sepsis, but that's probably not the real issue. It goes back to that. It goes back to that concept of selecting the right animals for the postmortem exam. These moribund animals are not going to be where it's at, but how do you find those ones that are just acutely ill before they're just lost all control to even close their shell?
Exactly, and that's where we need to have more money. I think that would help to pull people in. Money to try to tackle these big questions.
Certainly, and some of this sort of thing is really hard to get funded because I have this impression like that, these basic biomedical steps are kind of, they're cheap. They're not seen as like a new technology. They don't kind of garner a lot of excitement. But they're so necessary. And then the results are kind of seen as pilot work, but that's where we're at. That's the reality, is that in these sorts of investigations, it takes a long time to get some real theories going. So there is going to be a long pilot stage.
Michelle, that was fantastic. I have a question for you. You sort of indicated that somewhere around Spears Ferry, there was some challenges in terms of environmental disturbance. And I think you intimated that that may have something to do with mining. And were there events that occurred 2015, 2014, or even in 2016 that preceded the die-offs?
No, to my knowledge, there's an unknown event, any kind of, I'm trying to find that map, but to my knowledge, no, events are not known. This is just like for a long time, this area had historical coal mining. And the contaminants derived from that can stick around for, especially the organic pollutants can stick around in the sediment for a long time. I'm not aware of there being any, this is where you were talking about. I'm not aware of there being any kind of like dredging or activity that might have stirred things up. No, and all the reading I've done, I'm not aware of any kind of trigger. It just kind of came out of nowhere.
Thank you.
Great talk, Michelle. I did have one question about how we try to interpret what, the normal and abnormal findings are in these animals in the face of postmortem decomposition. Is that something that you struggle with a lot?
Yes, we want to avoid those ones at all costs, but it's a problem when you're sampling moribund and you can't actually be 100% confident that it's dead versus just moribund. We kind of use like the smell factor to decide for sure it's dead because it smells so bad. So, and they decompose super fast. So once they're dead, they are not worth sampling because they're going to decompose so quickly. Like in that aquatic environment, it's like being in a soup of bacteria, and that putrefaction of the tissue is the major part of the decomposition process. So that's another challenge. So many challenges, so little time, but actually sampling sick animals is pretty labor intensive to be out there looking for them and finding them when they're sick before they die. Once they're dead, they're no good.
In the chat, Sandra had said she wondered about long-term strip mining. And Tom Pavel asks, is it unethical to euthanize a moribund mussel?
Michelle,
did you?
yeah, sorry, we can't hear you right now.
Can you hear me now?
Yes.
Okay. It's not considered unethical as long as a humane technique is used, and the Association for the American Vet Medical Association has put forth some standards for humane euthanasia that are starting to include invertebrates. But specifically for mussels, it's a little bit more challenging because they conceal their shell, and any kind of anesthetic we immerse them in, they could just seal themselves off from it, and so it doesn't work. So we found, and also colleagues working in the area have found that a rapid freezing technique tends to be the most humane way. So that's it's not humane to just like take a mussel and dump it into some formalin for, alive, for waiting for pathology, nor will it give you good results, again, because they'll seal themselves off and they won't fix. So it's not unethical in the context of any mortality investigation as well, because you got to look at it from the perspective of we need these animals to figure out how to save the other ones. Okay, so I wonder if long-term strip mining, and I don't know the answer to that, Sandra, because I'm just not familiar with enough about the history of that area in Virginia. So I haven't dug into that. I've been learning so much, and that's one area I need to know more about.
Yeah, Southwest Virginia has quite a bit, or did quite a bit of deep mines as well as strip mines, and a couple of prisons were actually built on old strip mining sites.
There's definitely legacy contaminants in that area. So it's not going to be a simple, we're probably not going to find like a simple smoking gun of one single thing. There's probably going to be, you know, there could be one major thing driven by lots of other environmental issues, like a perfect storm.
Any other questions? I keep talking to, I forget I muted myself. Well, with that, thank you, Michelle, everybody. That was really awesome. Really appreciate it.